Hello 4620 and welcome to my video presentation. My project is titled SARS-CoV-2 Spike Protein, Receptor Binding Domain, and ACE2. The ongoing COVID-19 pandemic started with transmission of the novel and highly pathogenic coronavirus, Severe Acute Respiratory Syndrome Coronavirus 2. The initial outbreak of the virus has been traced to Wuhan City, China, back in December of 2019. With the growing number of cases and limited treatment options, the ongoing COVID-19 pandemic has put a strain on the economy and healthcare systems globally. Some devastating international and national COVID-19 statistics are listed here. COVID-19 research is accelerating to improve patient outcomes, reduce stress on hospitals, and to allow us to return to life as usual. SARS-CoV-2 is a spherical enveloped virus with a positive strand RNA genome. SARS-CoV-2 belongs to the beta coronavirus genus, a family of viruses that includes related SARS-CoV and Middle East Respiratory Syndrome coronavirus. All coronaviruses have envelope-embedded spike proteins, which mediate the attachment of the virus to its human cellular receptor. For SARS-CoV-2, this interaction occurs with human angiotensin-converting enzyme 2. The spike protein is a homotrimeric glycoprotein, with each monomer composed of an S1 and S2 subunit. The amino-terminal S1 subunit contains the receptor binding domain, or RBD, which is important for receptor recognition and attachment. The carboxy-terminal S2 subunit contains the hydrophobic fusion loop, which is critical for viral and host membrane fusion. The SARS-CoV-2 RBD has a twisted 5-stranded antiparallel beta sheet with short connecting alpha helices and loops that form the core of the structure. Between the beta 4 and beta 7 strands, there is an extended insertion containing the beta 5 and beta 6 strands, alpha 4 and alpha 5 helices, and loops. This extended insertion is the receptor binding motif, or RBM, which contains residues that directly interact with the ACE2 receptor. Human ACE2 is an enzyme expressed on the membranes of many cell types around the body, including lung alveolar epithelium. ACE2 lowers blood pressure by catalyzing the degradation of angiotensin II to angiotensin 1,7, a vasodilator. ACE2 counteracts the activity of angiotensin-converting enzyme, which produces the vasoconstrictor angiotensin II. Now let's look at the viral internalization and replication pathway. The S1 subunit of the spike glycoprotein binds to an ACE2 receptor on the alveolar cell membrane, initiating endocytosis of the virus receptor complex. In the endosome, the S1 subunit dissociates with ACE2 through cleavage of the spike protein at the S1-S2 boundary by a host furin-like protease transmembrane protease serine 2, or TMPRSS2. The S2 domain then transitions to a conformation that induces fusion of viral and host membranes. The coronavirus RNA genome is released into the host cytosol, where it is replicated by viral RNA-dependent RNA polymerase. The genome is translated into viral proteins by host ribosomes. These viral proteins assemble around the newly synthesized viral genomes to produce virions, which are released from the cell. Shown here is the interaction between the receptor binding domain of the S1 subunit and the human ACE2 receptor. ACE2 is shown in green, while the RBD core is shown in cyan and the RBM in red. The extended RBM in the SARS-CoV-2 RBD contacts the bottom side of the small lobe of ACE2, with a concave outer surface in the RBM that accommodates the N-terminal helix of the ACE2. Multiple tyrosine and other hydrophilic residues are involved with the interaction between the RBM and the N-terminal helix of ACE2. ACE2 receptor blocking may not be an appropriate drug target against COVID-19 because the loss of ACE2's cardioprotective effects can worsen disease progression. Instead, I would suggest two different approaches to preventing viral binding and entry: SARS-CoV monoclonal antibodies and TMPRSS2 inhibitors. Antibody treatment involves cross-neutralization of SARS-CoV-2 with a SARS-CoV monoclonal antibody. SARS-CoV and SARS-CoV-2 spike proteins have high sequence and structural similarity. SARS-CoV antibodies isolated from previously infected humans could be administered to bind to the receptor binding domain of SARS-CoV-2 spike protein, blocking its interaction with ACE2. However, improvements need to be made to these antibody therapies because many of them have failed to show efficacy. I would suggest targeting the unique lysine residue in the RBD of SARS-CoV-2 that is absent in SARS-CoV. This lysine is responsible for the more positive surface electrostatic potential in SARS-CoV-2 RBD compared to SARS-CoV. By substituting specific residues in the variable region of SARS-CoV antibodies to negatively charged residues like glutamic or aspartic acid, we could increase the affinity of these antibodies for SARS-CoV-2 RBD. TMPRSS2 inhibitors block the endosomal activation of spike proteins. Bromhexine is a plant-derived mucolytic that has been proposed for use against both influenza and coronavirus infections. Bromhexine interacts with residues within the active pocket of TMPRSS2, including glutamine 438, inhibiting its protease activity and thus reducing its activation of SARS-CoV-2 spike protein. Thank you for watching my presentation.